Good afternoon, guys. This is Chennavakarasu Harni. Today we're gonna say about a pharmacology. What is meant by pharmacology and what they are. Now, let us discuss about pharmacology. They can be defined as a study of substances that interact with the living system through chemical process, especially by binding to regulating molecules and activating or inhibiting the normal body processes. Medical pharmacology is often defined as a science of substance used to prevent, diagnose, and treat the disease. Toxicology. This is a branch of pharmacology that deals with undesirable effects of chemicals on the living system from individual cell to humans to the complex ecosystem. The actions of chemicals can be divided into two large domains. The first is the medical pharmacology and the toxicology, which is aimed at understanding the action of drugs as chemicals on the individual organisms, especially humans and the domestic animals. Both beneficial and the toxic effects are included. Pharmacokinetics. This deals with the absorption, distribution, and the elimination of drugs. Pharmacodynamics. They concerns the action of chemicals on the organism. The second domain, right side, is that of the environmental toxicology, which is concerned with the effects of chemicals on all organisms and their survival in groups and as species. The history of pharmacology, materia medica, the science of drug preparation and the medical uses of drugs began to develop as a precursor in pharmacology. Francis Magendie and his student Claude Bernard began to develop the methods of experimental physiology and pharmacology in the late 18th and the early 19th century. Advances in chemistry and the further development of physiology in the 18th and the 19th and the early 20th centuries laid the foundation needed for understanding how drugs work at the organ and the tissue level. Controlled clinical trial were introduced into medicine only about 60 years ago. Did it become possible to accurately evaluate therapeutic claims? Around the same time, information accumulated about the drug action and the biologic substrate of that action, the drug receptor. Studies of the local molecular environment of receptors have shown that receptors and the effectors do not function in isolation. They are strongly influenced by other receptors and by the companion regulatory proteins. Pharmacogenomics. The relation of the individual genetic makeup to his or her response to specific drugs. This is close to becoming an important part of the therapeutics. And the next one is discovery that small segments of the RNA can interfere with the protein synthesis with extreme selectivity has led to the investigation of small interfering RNA, that is siRNA, and microRNA, we can call it as miRNA, as the therapeutic agents. Similarly, short nucleotide chains called antisense oligonucleotides, synthesized to be supplementary to natural RNA or DNA, can interfere with readout of genes and the transcription of RNA. These intracellular targets may provide a major wave of advances in therapeutics. Two general principles that the student should remember is, the first one is that all substances can under certain circumstances be toxic, and the chemicals in botanicals, that is herbs and the plant extracts, are no different from chemicals in manufactured. Let us discuss about the introduction to pharmacology. The first one is nature of drugs. Drugs except for much greater proportion of impurities in botanicals. That all dietary supplements and all therapies promoted as a health enhancing should meet the same standards of efficiency and safety as conventional drugs and the medical therapies. There should be no artificial separation between scientific medicine and complementary medicine or alternative medicine. Ideally, all nutritional and botanical substances should be tested by the same randomized controlled trials as synthetic compounds. What are the general principles of pharmacology? That is, the drug may be defined as any substance that brings about a change in the biologic function through its chemical action. In most cases, drug molecule interacts as an agonist, that is activator, or antagonist or inhibitor, with a specific molecule in the biological system that plays a regulatory role. This target molecule is called a receptor. In a very small number of cases, drugs known as chemical antagonists may interact directly with other drugs, whereas few drugs, osmotic agents, that interact almost exclusively with water molecules. Drugs. They may be synthesized within the body, called as hormones, or may be the chemicals not synthesized in the body, that is xenobiotics. And the next one is poisons. Poisons, they are the drugs that have almost exclusively harmful effect. One of the famous scientists said Paracelsus. His name is Paracelsus. From 1493 to 1541, who lived in between that, stated the famous statement, "The dose makes the poison," meaning that any substance can be harmful if taken in the wrong dosage. Toxins. They are usually defined as the poisons of biologic origin synthesized by the plants or animals, in contrast to the inorganic poisons such as lead and arsenic. The physical nature of drugs. Drugs may be solid at room temperature, example aspirin and atropine, whereas drugs at liquid temperature is nicotine and ethanol, and the gaseous is nitrous oxide. These factors often determine the best route of administration. While speaking about the drug size, the molecular size of a drug varies from very small to very large. For example, lithium ion, their molecular weight is 7, whereas a protein molecular weight is 59,050. However, most drugs have the molecular weights between 100 and 1000. To have a good fit to only one type of receptor, a drug molecule must be sufficiently unique in shape, charge, and other properties to prevent its binding to other receptors. To achieve such selective binding, it appears that a molecule should be in most cases be at least 100 molecular weight units in size. Drugs much larger than molecular weight 1000 do not diffuse readily between the compartments of body. While speaking about the drug's reactivity and drug receptor bonds, drugs interact with the receptor by means of chemical forces or bonds. These are of the three major types of bonds. One is the covalent, another one is electrostatic, and the last one is the hydrophobic bond. Covalent bonds, they are very strong and in many cases not reversible under the biologic condition. For example, the covalent bond formed between the acetyl group of acetyl salicylic acid, that is aspirin, and the cyclooxygenase, its enzyme target is platelets, is not readily broken because of its size. Well, electrostatic bonding. This is much more common than the covalent bond in the drug receptor interaction, but are much weaker than the covalent bond. They vary from the relatively strong linkages between the permanently charged ionic molecule to weaker hydrogen bonds and very weak induced dipole interactions such as van der Waals forces. Here, let us speak about the drugs that bind through weak bonds to the receptors that are generally more selective than drugs and bind by means of a strong very strong bonds. This is because weak bonds require a very precise fit of the drug to its receptor if an interaction is to occur. While speaking about the drug shape, the shape of a drug molecule must be such as permit binding to its receptor site via the bonds. Optimally, the drug shape is complementary to that of the receptor site in the same way that the key is complementary to lock. The phenomenon of chirality, that is stereoisomerism, is so common in biology that more than half of all useful drugs are chiral molecules. That is, they can exist as enantiomeric pairs. Enzymes, they are usually stereoselective. One drug enantiomer is often more susceptible than the other to the drug metabolizing enzymes. As a result, the duration of the action of one enantiomer may be quite different from that of the other. Similarly, drug transporters may be stereoselective. Rational drug design. This is the design of drugs that implies the ability to predict the appropriate molecular structure of a drug on the basis of information about its biologic receptor. A few drugs now in use were developed through the molecular design based on the knowledge of the three-dimensional structure of the receptor site.